Thank you, Marcus. Really excited to be here to share the results from a recent collaboration with biocrates that we did to kind of do deep profiling of the plasma metabolome in people with HIV to investigate mechanisms of chronic inflammation. So in our lab, we kind of study the intersection of microbial dysbiosis, immunity and inflammatory responses, and systemic inflammation, and kind of how those three things come together to drive outcomes in infectious diseases like HIV and COVID-19, as well as women's health and cancer. Um, so antiretroviral therapy, or ART, is really good at treating and preventing HIV infection. We can see that HIV infections and related deaths have decreased steadily since about 2010. New infections are down about 39%, and related deaths are down about 51%. And the number of people living with HIV globally has actually been increasing steadily. So we have more people with HIV now than ever before. And this number is about 25% up since 2010. And this is mostly due to the increased effectiveness and access of antiretroviral therapy. But despite the effectiveness of ART, people with HIV continue to have chronic inflammation. And this leads to the development of inflammatory-mediated comorbidities, especially cardiovascular disease and cancer, which are some of the leading causes of death in people with HIV. So we can see here that having HIV comes with about a 2-fold greater risk of cardiovascular disease and up to a 19-fold greater risk of certain cancers like anal cancer. And this is even with antiretroviral therapy. The factors that are believed to contribute to this risk they're multifactorial, but chronic inflammation, as I mentioned, is one of them, as well as antiretroviral therapy. Just the drugs that these individuals have to take basically for the rest of their lives come with negative side effects, as well as dyslipidemia and substance use, which are more common in people with HIV. So furthermore, immunological non-responders are people with HIV who have poor T cell recovery despite taking antiretroviral therapy. They represent about 20% of the HIV population, and they're at a greater risk for developing some of these morbidities and at a greater risk for mortality. It's thought to kind of be driven in part by several different mechanisms. Gut dysbiosis and some of the inflammation that comes with that is one, as well as co-infections with viruses such as cytomegalovirus, dysfunctional and exhausted T cells, as well as just a decreased production and proliferation of CD4 T cells and increased apoptosis, leading to an imbalance of T cell subsets. So there's currently no interventions for improving immunological non-response. And kind of one of the focuses of our study is to look for ways of mitigating chronic inflammation in immunological non-responders, which could potentially reduce the occurrence of inflammatory comorbidities and mortality. So as a study overview, we enrolled 124 individuals from Mexico and Minnesota. 19 of those individuals were HIV negative controls. And then we had 105 HIV positive individuals. 69 of them were classified as immunological responders and 27 as non-responders based on their T cell criteria. And those individuals are all on antiretroviral therapy. We also had 9 HIV viremic individuals who were not on ART at the time of enrollment. We collected several different data types, including plasma metabolomics using the biocrates MxP Quant 1000 kit, as well as plasma proteins, including cytokines and gut barrier integrity markers. And we also measured the microbiome in rectum and ileum biopsies using 16S sequencing. So just an overview of the plasma metabolomic analysis. So we started with 1,231 metabolites from the MxP Quant 1000 kit. We filtered metabolites that had less than a prevalence of 20% in our samples, which gave us 697 metabolites to work with, which was pretty good because we had to dilute our plasma to inactivate it before being used. We then imputed values below the limit of detection, and then we calculate these metabo indicator features that we've mentioned today that are sums and ratios of the metabolites in our data set. So we ended up with 934 total features going into analysis, which we did unsupervised clustering, multiple linear regression, supervised clustering, pathway analysis, and multi-omic integration. When we look at the features that we had in this analysis, about 50% of them were lipids with 465 lipids, and then about an even breakup of 25% metabo-indicator variables and small molecules, respectively. When we kind of look further into the subcategories of these features, triglycerides dominated, or at least were the top class with 116 different triglycerides in our data set. And we also saw good representation of other molecules that we wanted to see, such as fatty acids and bile acids, which are important in our microbiome analysis. So now getting into some of the data. So this is a unsupervised clustering, or PCA, of kind of just the overall metabolomic data. And really kind of what we see is that the ART-treated groups, the immunological responders and non-responders and HIV negatives, really kind of clustered right on top of each other. You can see that their centroids are very close, whereas the viremic patients kind of clustered separate, further away from that primary group. So primarily our group's separated by viremic status, and kind of this is generally a good thing. It shows that the antiretroviral therapy does a really good job of restoring kind of an HIV negative phenotype for ART-treated patients. When we start to look at the pairwise comparisons between groups, here we use multiple linear regression to adjust for factors such as age, sex, years living with HIV, and country of origin. We kind of, as expected, see most of our large differences between our HIV negative and ART-treated groups versus viremic individuals, as shown in this heat map, which shows the log2 fold changes of those specific group contrasts. And we can see that the largest changes are primarily versus the viremic patients. But we do see some kind of more subtle differences between our HIV negative and ART-treated groups. When we start to look at the actual features that were different in here, now looking at differences between HIV negative and viremic individuals, kind of the two major things that stand out with these two groups was that HIV positive viremic individuals were really depleted of amino acids and phosphatidylcholines. Histidine being one of the major amino acids that was depleted in viremic patients. Another feature that we wanted to see was IDO activity, which Elise mentioned. And so IDO activity is one of the metabolo-indicator features. That's the ratio of kynurenine to tryptophan. And this was nice for us because it's kind of a positive control that's been discussed in HIV literature a lot as being increased in HIV positive individuals. So this represents the activity of the IDO enzyme, which catalyzes first rate limiting step of tryptophan metabolism. It indicates that pro-inflammatory status of viremic individuals. But what's nice is that we don't see it increased in the ART-treated individuals, kind of suggesting that mechanism of inflammation is decreased in our ART-treated groups. IDO activity has also been shown to impair T cell function, and it has immunosuppressive properties that HIV and cancer take advantage of to kind of avoid the immune system. When we looked at our ART-treated groups, so our immunological non-responders versus our responders, kind of the primary difference that we saw here was that dicarboxylic acids were higher in the immunological non-responders than the responders. And this is subaric acid, glutaric acid, and adipic acid. And so why this is kind of interesting to us is that this suggests an energy imbalance in immunological non-responders and really mitochondrial dysfunction. So these metabolites are all related to mitochondrial energy production. We know that carnitine is vital in mitochondrial metabolism, and we also saw that carnitine was slightly decreased in the immunological non-responders. Carnitine transports fatty acids into the mitochondria where they're used for energy. And so some of these dicarboxylic acids accumulate in the mitochondria and in the plasma when carnitine is deficient. So glutaric acid will accumulate when carnitine is reduced due to reduced clearance. And then subaric acid and adipic acid will accumulate due to decreased beta oxidation and increased omega oxidation of fatty acids. So we already know that carnitine deficiency is more prominent in people with HIV, but it's been less well known that it's actually distinguishable even between immunological non-responders and immunological responders. So addressing this energy imbalance in immunological non-responders may be one of the ways that we can help improve T cell function and reduce comorbidities in people with HIV. Next, we performed supervised clustering using a sparse partial least squares discriminant analysis. And this really also highlights that the mitochondrial features were the most important in separating our groups. See that glutaric acid, subearic acid, and adipic acid here were the most important. Another way to look at this analysis is to look at features that increase with disease severity or decrease. So biotin is one of these features that decrease with disease severity. We see that it was lowest in viremic individuals compared to HIV negative. We know that biotin or vitamin B7 is a cofactor for carboxylase enzymes. It's important for lipogenesis, fatty acid metabolism, and amino acid metabolism. We've seen that all of these are affected in HIV infection. Next, we did a pathway analysis, and we see that amino acid related pathways are impacted in both viremic and ART-treated patients. Alanine and biosynthesis of amino acid pathways were especially enriched in HIV-negative individuals, whereas phenylalanine, tyrosine, and tryptophan pathways were enriched in viremic patients. We also saw that arginine and proline metabolism was reduced in the ART-treated compared to HIV-negative groups, which is suggestive of an impact of ART therapy. So now we wanted to kind of integrate with our other omic features, so especially the cytokine variables and the gut microbiome, and kind of how all of these come together to mediate chronic inflammation. So when we look at the plasma features, really the largest difference we saw here was that immunological non-responders had increased IL-6 and increased IL-10 in the plasma, which traditionally are pro-inflammatory and anti-inflammatory cytokines, which we believe is suggestive of the chronic inflammation that these individuals experience. We also see in the microbiome that especially commensal bacteria such as Akkermansia and Bifidobacterium are depleted in the immunological non-responders, as well as Ruminococcus and L-stipes, which are all important short-chain fatty acid producers that are known to be important in gut barrier integrity. So when we do correlations between the microbiome and our cytokines, one of the interesting things that we saw was that Akkermansia and mucinophilia abundance was negatively correlated with IL-1 beta, IL-6, and the gut barrier integrity biomarker IFAB-P. So this is interesting because Akkermansia is kind of this new next generation probiotic that's been repeatedly associated with increased gut barrier integrity, anti-inflammatory effects, and metabolic improvements. So its depletion in immunological responders potentially reflects another mechanisms that we can improve chronic inflammation in non-responders. And kind of lastly, so network analysis here showing the correlations between all of our different data types and the plasma metabolome from the MxP Quant 1000 kit. We see here is all the correlations with edges that are blue representing negative correlations and red edges representing positive correlations. And really the most striking difference that we see here is that IFAB-P, or this gut barrier damage biomarker, was really the most connected to the plasma metabolome. You see a lot of different positive and negative correlations with these metabolites, suggesting that gut barrier integrity is really important in kind of influencing the plasma metabolome. We also see a couple notable correlations with IDO activity and LBP, which is a marker of microbial translocation, so as IL-18, which is kind of involved in gut barrier homeostasis in mucosal surfaces. And we only observed a few correlations with microbiome taxa and some of the metabolites, but especially with commensal bacteria like Doria longicatina and phenylacetate acid, which was increased in our viremic individuals. So in conclusion, antiretroviral therapy does a really good job at restoring the plasma metabolome back to kind of an HIV negative phenotype. We saw that the metabolome of viremic patients was highly disrupted primarily depleted of amino acids and enriched for tryptophan metabolites. We saw that kind of the differences between immunological responders and non-responders were subtle, but mostly in metabolites related to fatty acid and mitochondrial metabolism. We think that interventions, including carnitine or biotin supplementation in immunological non-responders, could be interesting avenues to pursue to mitigate chronic inflammation. We also see that gut barrier damage marker IFAP was highly correlated with the plasma metabolome, and interventions looking at improving gut barrier strength, potentially through probiotics, such as with Akkermansia mucinophilia, could also be used to mitigate inflammation in immunological non-responders. And then lastly, just some closing thoughts on using the MxP Quant 1000 kit here for our analysis. So the data return from biocrates was really fantastic, and it made it really easy to go directly into analysis. This includes like really good feature labeling and metadata. They provide all the HMDBs for all the metabolites, which makes it really easy to do pathway analysis quite quickly. And also really great quality control. So you can be really confident in using your data and inferring from the biology there. The metabo indicator features also cover a really impressive number of biologically informative sums and ratios. And they provide a nice Excel spreadsheet that has references to literature and annotates all of them, so you can easily kind of gain more biological knowledge out of those ratios and sums. And then there's also really great coverage of metabolites that we wanted to see, such as short-chain fatty acids and bile acids, which are really relevant to microbiome research. And this was a big improvement over the previous kit we were using, which was the MxP Quant 500 kit. And then lastly, the lipid coverage was also really great. We are less kind of used to lipids in our own analysis in the lab, but a lot of the interesting features that showed up in this analysis were lipid related. And lipids are being increasingly recognized in HIV research. So it was really nice to have this big reservoir of lipids to look at. So with that, I'd just like to thank my lab, Dr. Nicole Klatt's lab at the University of Minnesota, as well as Tim Shacker's lab at the University of Minnesota for helping to procure and provide the support for this research. So also a big thank you to biocrates for this collaboration.